Hello, my name is Catherine Métayer. I am faculty at the School of Public Health, University of California, Berkeley, and I am the director of the Center for Integrative Research on Childhood Leukemia and the Environment. The goal of my research is to better understand what causes leukemia and other cancers in children. There are many ways parents and children are exposed to pesticides, whether it is using pesticide at home, at work, or living in proximity to farming and public spaces where pesticides are applied. All those sources are increasing chances to track pesticide indoors, where young children spend most of their time and can be exposed by touching, ingesting, and inhaling these chemicals. Because of the various ways we can be exposed to pesticides, it is challenging to assess what types and how much one can be exposed to. This is why researchers have used different methods like conducting interviews, linking addresses at residence to publicly available databases that collect information on agricultural pesticide use, or collecting home dust that may contain many persistent chemicals such as pesticides. In contrast, biomarker studies are rare because of the difficulty to collect blood and urine samples when studying relatively rare diseases like childhood cancers. This graph shows how much pesticides are being used worldwide, with Brazil and the United States being the top two countries using agricultural pesticides. California is the largest and most diverse agricultural producer in the United States, producing more than half of the country's fruits, vegetables, and nuts, and using approximately 22% of the total agricultural pesticide in the nation. We conducted a study in Northern and Central California, where the majority of farm workers are Latinos. Working with industrial hygienists and occupational epidemiologists, we collected information on the type of jobs and tasks from the parents and assessed their potential exposure to pesticides. Fathers who were exposed to pesticide before or after the child's birth had a 50% increased risk of having a child with acute lymphoblastic leukemia. No increased risk were reported for maternal exposures, with odds ratios close to or overlapping 1. Although much fewer mothers were exposed to pesticide at work, limiting our ability to detect an association. Because our California study was relatively small, we collaborated with other childhood leukemia studies around the world that are part of the Childhood Cancer and Leukemia International Consortium. This pooled analysis of about 8,000 cases and 14,000 controls using interview and expert assessment confirmed our finding from California. We examined more closely the type of crop being produced and found that fathers working in the production of nuts had the largest increased risk of having a child with leukemia. The production of nuts in California has increased in the past years, and it should be noted that harvesting, transporting, and processing nuts generates a lot of dust, substantially increasing exposure pathways via inhalation, ingestion, and skin contact, even more so if protective equipment is not being used properly. Communities living in proximity to agricultural pesticide use are particularly vulnerable. As mentioned earlier, Brazil is the top user of agricultural pesticides in the world, and researchers have shown that the intensification of soy production from 2004 to 2019, as illustrated on this map, was associated with an increased risk of childhood lymphoblastic leukemia. Similarly, a recent study from Nebraska in the United States has identified several pesticides, mostly herbicides, linked to increased risk of childhood cancer overall, especially leukemia and brain tumors. Using any type of pesticides at home before and after the child's birth has also been found to increase the risk of acute lymphoblastic and myeloid leukemias. These analyses were done by the Childhood Cancer and Leukemia International Consortium using large sample size. While leukemia is the most common cancer in children and the most studied, other childhood cancers have been associated with exposure to pesticides, including brain tumors. As shown from the meta-analysis of published data from various countries, the magnitude of the association ranged from 17% to 32% increased risk. Studies in several countries showed that occupational or residential exposure to pesticide also increased the risk of Wilms tumors, a type of kidney cancer in children. Same findings are found for lymphomas. Our group took a complementary approach to characterize a child's exposure to chemicals, including pesticides, by collecting dust in the homes of families participating in the California childhood leukemia study. Dust is a major exposure pathway for young children, and because it is a reservoir of semi-persistent and persistent compounds, it is a good medium to estimate past exposures. We measured dozens of pesticides and found that levels of herbicide dactyl, especially when used with allochlor, increase the risk of acute lymphoblastic leukemia in children. We, however, did not detect association with any other pesticides. We know that pesticides are found everywhere in our environment. Meta-analysis of self-reported use of pesticide at home, at work, and residential proximity to pesticide support its association with increased risk of several childhood cancers. Indirect assessment of exposure to pesticide in epidemiologic studies can be imprecise and underestimate true associations. Biomarker studies are rare due to the difficulty to obtain pre-diagnostic biospecimen.